Is there like a structure he did there?
No. I did point though.
All right, good afternoon, everyone. I'm very happy to introduce my lab mate and one of my closest friends, Lindsey, who's joined the lab when I did about 3 years ago. We both work with Scott, but it hasn't been known. And she's going to be presenting on some work she's done in collaboration with not only Scott, but a group over at Cornell, which is really cool. And she's currently in the process of deciding whether she's going to stay here next year, play basketball, and do some more work with Cornell, or go get a cool job in industry. So without further ado, Lindsey Albertelli. Great. So my project is the reassessment of glutaminase inhibitor compound 968. The reassessment part will make sense at the end, but compound 968 is an anti-cancer compound that targets the metabolism of cancer cells. So I'm going to go into a brief background of the altered metabolism of cancer cells. So as you may know, cancer cells grow and divide at a faster rate than healthy cells. And with that, Otto Warburg termed a sort of a classification of this altered metabolism called the Warburg metabolism or the Warburg effect. And there's kind of three main points with this. And the first is that cancer cells have increased glycolysis. So the glucose to pyruvate is occurring at 200 times faster rate in cancerous cells compared to healthy cells. And with that, cancer cells also have aerobic lactate production. So they are converting pyruvate to lactate even in the presence of oxygen. In healthy cells, this only happens in anaerobic conditions. So when there's no oxygen, and you may associate this with lactic acid production in your muscles, and like that's sort of what makes you sore. And the third is glutamine addiction. So glutamine is just found in your body, it's an amino acid, and cancer cells can uptake this and use it as a source of fuel. And glutamine is converted to glutamate, which is then converted to alpha-ketoglutarate, and this serves as an entry into the Krebs cycle. And a little bit deeper into this pathway, glutamine is deaminated into glutamate by an enzyme called glutaminase, which is then further deaminated into alpha-ketoglutarate by an enzyme called glutamine dehydrogenase. And researchers have found that if you inhibit glutaminase, so if you inhibit the conversion of glutamine to glutamate, this actually serves as an anti-cancer therapeutic. And it does this by preventing, kind of cutting off a source of fuel for those cancer cells so they can no longer grow and divide. And so there are some known glutaminase inhibitors. BPTS is kind of the most widely known inhibitor, and this is its crystal structure. In the blue and the teal is the glutaminase, and then the white compound in the middle is BPTS bound to glutaminase. And then CB839 is actually a derivative of BPTS that's in clinical trials right now as an anti-cancer therapeutic. Compound 968 is the compound that I worked with. It was found as a glutaminase inhibitor by the Sarione lab at Cornell. And one of the ways they showed it was a glutaminase inhibitor was through agar experiments. And so, as you know, cancer cells can grow and divide on soft agar because they don't need to be anchored to anything, whereas healthy cells don't need to be anchored. And so you can see the colony formation in the control on the soft agar. And then you can see when you treat it with 968, that colony formation kind of disappears. So it's showing it's an anti-cancer therapeutic. And so my project originally started out with as creating derivatives of compound 968. And that's the Sarione lab reached out to us and asked us to do this for them. And this is the reaction used to make compound 968. In the red, we have an aldehyde. The pink is naphthylamine, and the blue is dimidone. And you basically add these all into a rad-bound flask, heat and stir for a couple hours, and then you should get compound 968. But we came across some literature that actually suggested that this reaction does not make 968. Rather, it makes an isomer of 968. You can see the blue and red groups are switched. And so this summer, we basically proved that 968 is actually follows the corrected structure rather than the structure that it's advertised as. And so for the rest of the presentation, you will see compound 968 referred to as its corrected structure. And if you have more questions about this at the end, I'd be happy to answer them. But for time purposes, we'll just keep moving. And so once we've had created analogs this summer, we wanted to start testing them. And we used this glutaminase assay, which is a coupled assay. And the first part of the assay is the glutaminase reaction. So glutamine is deaminated to glutamate. And this is where the inhibition happens. So BPTS and 968 are inhibiting glutaminase. And then glutamate is further, in the secondary reaction, glutamate is converted to alpha ketoglutarate by an enzyme, glutamate dehydrogenase. Subsequently, NAD+ is reduced to NADH, and that is what we measure in the spectrometer. So we first tested BPTES, and on the y-axis we have GAC percent activity. GAC is the same as GLS, so same as glutaminase. And then the concentration of BPTES on the x-axis. And you can see that as the concentration of BPTES increases, glutaminase activity decreases. And it has an IC50 of a little less than 100 nanomolar, so it's a very potent compound. And this is a dose response curve that you would expect for a normal drug that has a specific binding pocket. When we tested compound 968, we didn't really see what we expected. So first, you can see there's barely any inhibition of glutaminase by compound 968 when it's reported to have an IC50 of around 10 micromolar. So this was very abnormal, and we were sort of like, what's going on? And so we lowered the concentration of glutaminase to 5 nanomolar, and we increased the incubation time. So we increased the time that we allowed the enzyme to interact with 968 before initiation. And you can see we got inhibition, but the dose response curve showed a very steep drop off known as a steep hill slope. And this sort of raised some red flags for us. So throughout all of our time with compound 968, Scott and I developed a list of these peculiar characteristics of compound 968. First, it requires pre-incubation time. It's inhibition sensitive to enzyme concentration, and it has a steep hill slope, as through this evidence showed right here. And so we had many conversations about it. We kept saying 968 is so weird. It's not functioning the way it should. It's not operating like a normal drug. And Scott kind of brought up this idea of a PAIN, or a pan-assay interference compound, also a non-specific inhibitor. And so I decided that after my thesis, I wanted to determine if compound 968 is a PAIN. And we came across this widely known paper that was published by Brian Chochette at UCSF. He is a big PAINs guy. He does a bunch of stuff with that. But he basically showed this a specific mechanism of non-specific inhibition. And I wanted to determine if compound 968 followed this specific mechanism of non-specific inhibition. And this specific mechanism of non-specific inhibition is known as aggregators or aggregation. And these aggregators form these particles in solution right here. And to me, they kind of look like micelles a little bit, but in solution. And then when you add the enzymes stick to the outside of these particles in a non-specific way. So any part of the enzyme can stick to the outside of that particle, and the particle also can stick many different types of enzymes. So it's non-specific. And once those enzymes are stuck to the outside of these particles, they can no longer catalyze reactions. And so this is abnormal. They don't really have biological activity usually, whereas a normal drug like BPTS has specific binding pockets, interactions with specific amino acids, and only inhibits glutaminase. And so to determine if compound 968 is an inhibitor, we first wanted to determine if compound 968 inhibits unrelated enzymes. And we did this by testing the inhibition of compound 968 against malate dehydrogenase and beta-galactosidase. And then after that, we wanted to determine if this enzyme inhibition is blocked by additives that affect aggregation. So we added different things that basically somehow perturb this gray sphere and then measured inhibition. So to start off, the enzymes we used, the first one is GAC, glutaminase, an amino acid hydrolase. And then we also use beta-galactosidase, which is carbohydrate hydrolase, and malate dehydrogenase, which is an alpha hydroxy acid oxidase. So if it's a normal drug, it should not inhibit beta-galactosidase or MDH. It should only inhibit GAC. If it's an aggregator, it will inhibit all three of these enzymes. And so we first tested it against glutaminase, and you can see inhibition. And then we tested it against beta-galactosidase, which it also inhibited. And then we tested it against malate dehydrogenase. And so we determined that it inhibited all three enzymes. And you can see it has an IC50 in all three of them, around 10 micromolar. And so from this, we were able to determine that compound 968 is a non-specific inhibitor. It can inhibit multiple different types of enzymes. And so our next question was to determine if if this inhibition is mediated through aggregation. And we started off by figuring this out with a detergent called Triton X100. And Triton X100 basically prevents the formation of these gray spheres. So it's thought if these gray spheres can't form and it's an aggregator, then the enzymes can't stick to the outside and there should be no inhibition. And so we tested it against glutaminase, and you can see in blue we have no additive, the normal inhibition. But then when we add triton, that inhibition goes away by that flat line showing no inhibition. And then we tested it against beta-galactosidase. The same thing happened. Inhibition went away by triton X100. And then we used malate dehydrogenase, which also went away with triton. So we determined that, so at this point we were like, yeah, like 968 is probably an aggregator, but we wanted more evidence. So we used a decoy protein experiment. And the way that this experiment works is if you add this 968 or an aggregator into solution, and then we add a decoy protein, which is BSA or bovine serum albumin. And what happens is BSA will coat the outside of the particle. So then when we add the enzyme of interest, glutaminase, then glutaminase cannot stick to the outside of this particle. So inhibition should go away if it is an aggregate. And so we first tested it against glutaminase. In the blue, we have no additive. In the red, we have the decoy protein. And you can see that inhibition goes away upon decoy protein addition. And then we tested against malate dehydrogenase. And again, you can see that inhibition goes away. And there was one more experiment that we could do that would basically confirm that 968 is an aggregator. And this was a centrifugation-based experiment. And the thought behind this is that compounds that form aggregates are centrifugal. So they will form pellets at the bottom of the centrifuge tube. Whereas normal compounds that don't form aggregates will not form a pellet at the bottom of the centrifuge tube. And so what we did is we did an assay. And for one of the conditions, we used the supernatant after centrifugation. So there should be no compound 968 in that supernatant that's added to the assay. So if it's an aggregate, then that inhibition should go away when that supernatant is added. So we did it against malate dehydrogenase, and in the gray, we have just normal malate dehydrogenase, no additive, normal, just control. And then in the light blue, you can see we added 968 that was not centrifuged. So at 10 and 50 micromolar, you can see inhibition. And then we centrifuge compound 968, so we only added the supernatant, we added this blue stuff, and you can see that that inhibition went away. And then another centrifugation experiment that we did involved putting not only the 968 into solution before centrifugation, but we added the protein. And so it's thought that these spheres with the protein on the outside will form a pellet at the bottom. And then if you take that pellet and then run it on a protein gel, then if that protein is brought down into that pellet, then it will show up on the protein gel. And so, but proteins themselves are not centrifugal. So if you just centrifuge protein in solution, there will not form a pellet of just protein at the bottom. So we ran this gel. First, we ran it with no 968 in solution. So we just added malate dehydrogenase to the centrifuge tube and centrifuged it. And you can see that there is no silver band. So no enzymes showed up in this lane. And then we did it with 968 with increasing concentrations. And we did it with malate dehydrogenase, and you can see that as the concentration of 968, that band gets darker and bigger, indicating that more malate dehydrogenase was pulled down, and that these little spheres were in this pellet, which were then added here, and the protein showed up on the gel. So just in contrast, CB839, a known drug, a known specific inhibitor that binds a specific pocket, we tested it against glutaminase. And you can see it inhibits glutaminase, but when we test it against malate dehydrogenase, there's no inhibition. It's a specific drug, a specific inhibitor of glutaminase. And we did these same experiments with CB839 with triton and BSA. And you can see that triton and BSA have little to no effect on the inhibition of compound 968. Sorry, the inhibition of glutaminase. And so that just shows, in conclusion, compound 968 is an aggregator. It inhibits unrelated enzymes, and this inhibition of these unrelated enzymes are based on the formation of this aggregate particle. And we basically showed this by perturbing that aggregate particle with triton, BSA, and centrifugation. So in all of my time here, if you look up 968, this is the structure. So this is the structure it's advertised as. This will probably be changing to the structure that we solved it to be. And its function as a glutaminase inhibitor is sort of under review. And so although its function as a glutaminase inhibitor is kind of thought to not be true, It does still have anti-cancer effects. So determining the target of compound 968 could be a very cool future direction. It might reveal some new biology about how it inhibits the growth and proliferation of cancer cells. So thank you to Scott, DJ and Robbie, DJ Robinson and Andy for being my committee this semester, Chun for solving the crystal structure and for everything she does in the department, the Serione Lab at Cornell for bringing this project to us, and everyone in the chemistry department. And I would also like to put out a special thanks to Janet for introducing me to Scott my freshman year and for just being such a great person and always being so helpful. Thank you. Any questions? Yeah.
Did the isomer develop only when you did the combination, or is the one from Sigma Aldrich also incorrect?
So this is sort of the evidence of how we figured it out. So the green one is 968. We bought it from Sigma and it's advertised as its known structure. We made 968 and then we also bought the isomer of 968 that's advertised as the isomer from ChemDiv, and they all have the same NMR, and then we solved the crystal structure of our synthetic material and it was the isomer. Okay, so this is NMR spectrum, and so you can see like all of the peaks are the same.
And what peaks would you expect to be around?
That's a great question that I don't know. Maybe Scott, do you know the answer to that?
Well, it would be different in some way.
Yeah, it would be different in some way. We don't know
Sometimes
exactly
having
what it would
unique
be.
spectrum.
Yeah, because there's no NMR of the other one.
You know why it's an aggregator? Like, what's the property that's causing this?
It's not really known what that property is. It's just the way it functions in the solution. Yes, ma'am.
Why had that?
No.
Did you do any like NOE experiments to see like which protons were close to each other? You know, like, okay.
We did carbon NMR and those were also the same. And we also tested them, all 3 different compounds against 968 and against contaminants, and they all have the same inhibitory capacity.
I noticed that this is a terminology thing that I don't know about. I noticed you referred to it as an anti-cancer therapeutic,
Yeah.
and it seemed like that was probably a deliberate choice of word. Is that different than a drug?
No, it's not different than a drug. I just referred to it as an anti-cancer therapeutic because in this sense, we kind of were showing that it's not actually a drug of contaminase. So that's sort of why I use those words. Yeah, Marky?
I just wanted, this is a little bit tangential to what you actually hit on, but are there any side effects of inhibiting contaminants that you don't get doing it?
They haven't actually found any side effects of that. Glutaminase, actually, they found that these compounds that inhibit glutaminase only work against cancer cells, so they have no effect on normal cells.
Yeah. Yeah, I think the clinical trials are ongoing, so that the data on like side effects.
Yeah, you would think, right?
Or maybe they have them both.
Yeah,